Everything I said was up on the board, except pancreas you guys didn't kind of figure out. Okay? So that's the control. Yes?
So basically just like what
does where? What does where? What stimulates it? So I will have parts of that information on there and you guys will fill in the rest. Okay? Sometimes I do it in table form, sometimes I do it like this and you guys draw the line.
So kind of like what stimulates what in what way?
What inhibits what and where does it come from? Okay, so what's the molecule will be on the diagram and then it has an effect on one organ by doing what? What causes, so, you know, what causes gastrin to be released, what causes GI to be released, what does it do once it's released, what organ does it bind to, and what's its effect. Okay?
So we'll have the molecule.
Yeah. All right, so now let's look at how, now that the chyme is in the small intestine and it's been mixed with bicarbonate ions, its pH has been changed, the pancreatic enzymes have been added, what do we do with digestion? So we're looking at carbohydrate digestion. So page 48. This diagram looks more complicated than it actually is. Except for the addition of the enzymes, this is what we talked about in the proximal convoluted tubule with the movement of glucose out of the filtrate into the blood. Same import type of movement, all right? But now we need to get those simple sugars. We're still at the disaccharide, polysaccharide state. So, can I erase all those colors on the board? I'm scared to do that after all that work, but.
You said this was on page 48 of the notes? The table's not there.
Not the table, where we're going to look at now at the digestion of carbohydrates.
Is it with the maltase?
Yes. All right, so we have the simple columnar cells. These are the cells that are on the villi. All right, so we're talking about the simple columnar cells that are lining, and well, not lining, but covering the villi. And I'm drawing the microvilli fatter than they actually would be. So this would be the lumen of the jejunum and ileum. And we're looking at carbohydrate digestion first. Right? So while we had a little bit of intermediate digestion with the salivary amylase, did any of that continue in the stomach? No. So we're going to have a lot of starches, all right? Long chains of sugars, so polysaccharides present in the lumen. And the enzyme that we have added via the pancreatic duct from the pancreas interacting with our polysaccharides is pancreatic amylase. That takes place in the lumen. So we'll write that over here. And we saw this same reaction when we were looking at salivary amylase. So we've already gone through this process with salivary amylase. Only now, instead of salivary amylase, our enzyme is pancreatic amylase. And our substrate is going to be disaccharides. All right? So we have 3 disaccharides that are produced by the enzymatic breakdown of polysaccharides. And those are sucrose, maltose, and lactose. Right? So these are disaccharides. Each circle represents a single sugar. So that's the product of this reaction, the intermediate stage of digestion, is the release of disaccharides. Are those small enough to cross the plasma membrane and move into the cell and into the tissues?
No.
No, so we need our final stage of digestion. That final stage of digestion is moderated by enzymes produced by the columnar cells of the small intestine, but not released into the lumen. These enzymes are embedded in the plasma membrane of the microvilli. And they are specific to disaccharides. So when the disaccharide interacts with this, it results in the separation of the bonds holding the two sugars together and the release of individual single monosaccharides. So if we take our monosaccharide, our disaccharide sucrose and maltose and lactose, each of these has a specific enzyme that it will only interact with. So the enzyme that interacts with sucrose is sucrase. Usually enzymes end in ASE, but not always. Pepsin and trypsin are enzymes, and they end in an IN. All right, gastrin is a hormone, and it ends in IN, so you can't use that as a complete, foolproof differentiation. Maltose interacts with maltase. You can see where this is going. And lactose interacts with lactase. Yes?
So the blue dots are the pancreatic enzymes?
Not the blue dots. The blue dots are the intestinal enzymes. They're produced by the simple columnar cell and embedded in the plasma membrane of the microvilli. So the disaccharide has to come very close to that simple columnar cell to interact with that enzyme. Once it does so, it splits that into a monosaccharide, and now that as a single sugar can enter into the cell and be absorbed. Okay? So the products of sucrose after interaction with sucrase is 1 glucose and 1 fructose. Now this is a monosaccharide. It's small enough to pass into the simple columnar cell. Maltose produces 2 glucose molecules. And lactose produces glucose and galactose. So remember, we use sodium as a symporter in the proximal convoluted tubule. And so here we have sodium with a co-transport, secondary active transport, moving these monosaccharides, glucose and galactose, glucose and fructose, into the simple columnar cell. Okay. And then it's variable whether secondary active transport or facilitated diffusion is used to exit the cell and move into the capillaries of the villi. You don't have to worry about which one is which for that exit. It's the same process that we looked at in the proximal convoluted tubule for moving the glucose from the filtrate to the peritubular blood, as we see here moving the glucose from the lumen of the intestine to the capillary blood of the villus. So this is the final stage. These enzymes are the final stage enzymes that provide an absorbable form of the sugar. Okay, now let's look at protein. In the mouth, we had no enzymes for protein. All right, so no digestion occurred. In the stomach, Our enzyme, when activated by hydrochloric acid, was pepsin, but it still just produced smaller peptides, not individual amino acids. So we had no absorption. And now that we are in the small intestine, we have enzymes trypsin, that should be a chymo, not a chyme, trypsin. And they'll do the same, very similar to pepsin. They'll produce smaller strands of amino acids, but not individual amino acids. So they're similar to the pancreatic amylase. So instead of being simple sugars, we're going to now call these circles amino acids. And instead of a starch of polysaccharides, we have the polypeptide of our protein. All right? And this enzyme is no longer pancreatic amylase, but trypsin and chymotrypsin. Pepsin is no longer active because of the difference in pH. Instead of producing all dipeptides, we'll have longer chains, but smaller than what they were originally. All right, so our polypeptide is going to be broken down into oligopeptides, which just means many, so, but like several rather than lots. So oligopeptides and some double ones. All right, so that again is taking place in the lumen of the small intestine, regulated by pancreatic enzymes. So we get smaller, but no single amino acids. Now, the pancreas also produces an enzyme known as carboxypeptidase, which the small intestine does as well. So it can remove individual amino acids. You can see there at the carboxyl group, single amino acids are produced, but not as many. Carboxypeptidase and aminopeptidase, in addition to that, are the enzymes produced by the small intestine. They are brush borders, so they are bound to the microvilli. So now instead of being sucrase and lactase and maltase, these blue circles represent aminopeptidase and dipeptidase. Right? So what happens is, with our oligopeptidide, instead of writing it out, I'm going to draw it out. We have a strand of amino acids. The amino acid on this end has the free nitrogenous or amine group. The amino acid on this end has the amino acid or carboxyl group. And so this is going to be worked on by aminopeptidase, whoops, wrong color, bound to the brush border. And it's going to interact right here between the two amino acids on the amine group, right? And it will produce a single amino acid, which will be absorbed by the small intestinal wall. But we still have the rest of the chain left. Okay? Then, that other chain with its carboxyl group has another nitrogen group here that's now exposed. And this time, the carboxypeptidase, and these can happen at the same time. I'm drawing them separately. Bound to the plasma membrane of the microvilli will work here. And will release another single amino acid. And now we're left with a 2 amino acid or a dipeptide, and that's where dipeptidase comes in. Okay? So you can see this on the screen. The carboxypeptidase is working on the carboxyl group, and the amino acid peptidase is working on the amine group. And we have individual amino acids, and then the dipeptidase comes in and works where we have just 2 amino acids left. And we don't have anyone that's only an amine end or only a carboxyl end.
So this is the sequence in which they work with the trypsin?
Well, they don't, both of these can happen at the same time.
Okay.
So the aminopeptidase can work here at the same time that the carboxypeptidase is working there. Okay?
And the trypsin will come in?
And well, trypsin is the one creating this, because it was like, you know, hundreds or 500 amino acids long. And trypsin will break it down into 10 or 20 or 40 amino acids. And then the aminopeptidase or the carboxypeptidase will release one single amino acid at a time from either end until we get to just 2, and then the dipeptidase works on those 2.
So technically both of the aminopeptidase and the
Carboxypeptidase?
Yeah. So both of them technically work in a little bit different ways to make amino acids.
Yeah. So they'll be bound to the plasma membrane, and depending on which end of the amino acid, the protein they come in contact with. So if the carboxypeptidase is coming in contact with this, it won't work. It has to be here for the carboxypeptidase to work. And the amino acid peptidase has to be here. Yes?
So the substituent groups of the amines and the carboxyl groups being split off and then rejoined and created by dipeptide? Is that
No, it's just I'm saying, we lost this one, and we lost that one, now we only have 2 left. And that's when the dipeptidase comes into play.
Oh, I get it, okay. So the rejoining of the 2
Nothing
groups of a
rejoins.
smaller chain just
I was just showing there, this one has a nitrogen group too.
They all do.
Oh, so
Each amino acid has a nitrogen group. Since this is a single amino acid, it has its own carboxyl end and its own nitrogen end.
Okay, I misunderstood what was taken.
Yeah, so I'm not rejoining it, I'm just saying, once this is free, we now expose
Okay,
and can write out that carboxyl,
got it, perfect.
okay? Again, it's a cartoon, it's not the direct chemical modeling of structures. So back to this diagram again, to reinforce that. So here we have 4 amino acids. All right, the carboxy group works on the end that has the carboxyl component, the amino acid, aminopeptidase works on that, and both of these again are in the microvilli. They're not mixed freely in the lumen of the small intestine. And then the dipeptidase splits the 2 remaining amino acids. So now we have 3 amino acids, and they are also, here's the different amino acids bound, enzymes bound to the microvilli. And this doesn't illustrate it the same as the carbohydrate one. But remember how amino acids were reabsorbed from the filtrate in the proximal convoluted tubule? What system did they use? It's diffusion. They go from a high concentration to low. They use the same type of symport, secondary active transport, as glucose did. Remember when I wrote it on the board, I wrote underneath it amino acids and other additional vitamins and other molecules. So even though it's not shown here, there would be the same type of sodium-driven secondary active transport and protein transport molecules creating channels that will move the amino acids across the cell and into the, again, vascular capillaries. So all those words and drawings look complicated, but if you just think of, you know, we go from long chains to short chains to individual amino acids released from either end, depending on the enzyme that's interacting with them. All right, so that brings us to lipids. Now, every area that we looked at had a lipase. So we had salivary lipase. Okay, we have gastric lipase. So for each of those enzymes, we were missing an important component, bile salts. And now in the duodenum, we've added the bile salts from the gallbladder, or new bile that was synthesized by the liver. Are bile salts an enzyme? No. Bile salts are not an enzyme. So if they're not an enzyme, how do they increase the rate of the reaction? Enzymes do so by binding to the substrate and kind of torquing the shape of it, making the bonds more likely to be broken or interact with water or whatever. In this case, it's water. It's going to break those bonds. So how do bile salts function to increase the rate of the reaction? Remember the surface area concept? They're a form of mechanical digestion. So they maintain the state of mechanical digestion that occurred in the stomach. All that twisting and churning was like taking oil and water or oil and vinegar in a salad cruet and shaking it, getting tiny little droplets suspended in the water. If you let it sit, those droplets bind each other because they don't like the water and they recoalesce, and the oil and the water separate. Okay, so we've suspended the oil and the gastric juices in the stomach, but as they move into the small intestine, they start forming larger and larger droplets. The bile salts function by coating them with a substance that is both water, that's like the plasma membrane itself. It has polar and nonpolar regions. The nonpolar regions interact with the lipid droplet, and the polar regions interact with the water. All right, and that helps to coat them and keep them from joining with additional lipid droplets. So this would be the hydrophilic end, and this would be the hydrophobic end. This is our lipid. All right, and that maintains the suspension or the emulsification of these small droplets and increases their surface area. So our enzyme is pancreatic lipase. The rate of the reaction has been increased because we're maintaining this larger surface area. All right, so we just have more surface area for the enzymes to interact. And we're just going to look at triglycerides. Okay? So triglycerides were formed of a glycerol molecule, which is 3 carbons, and then 3 fatty acids attached to those. And these fatty acids can be 8, 10, 20 carbons long. What I haven't put on there are the hydrogens, but there would be hydrogens present as well, because each carbon can form 4 bonds. So we'd have 2 hydrogens off of here. All right, so that's the glycerol. And each carbon of the glycerol molecule has a fatty acid. This is the substrate that we're going to be working with on Tuesday in our wet lab next week. So the effect of lipase is to break these bonds right here. So what we end up with, instead of a triglyceride, is a monoglyceride plus 2 fatty acids. That's the most common. Okay? Occasionally you might have just a free glycerol, or you might have a diglyceride. But this is the most common result. Monoglyceride and fatty acids. You guys remember how I said you would be able to tell that this happened on Tuesday's lab? We have cream that we'll mix with litmus powder. Litmus powder is purple at an alkaline pH and pink at an acidic pH. So what's going to happen to the pH as digestion occurs? It becomes acidic. So our purple cream is going, our purple cow is going to change to a mauve and then a pinker color as more and more fatty acids are released. Okay? Now, this starts to occur while it's still associated with bile salts. And so this cluster of triglycerides and monoglycerides and fatty acids associated with the bile salts is known as a micelle. If you have a daughter, Diane, you can call her micelle.
That's my chef.
Now, lipids are easily absorbed across the plasma membrane of the cell because it's a lipid, all right? So, what we have then is the micelles come in contact with the microvilli. We don't have enzymes there because we don't need another final stage enzyme by the small intestine. It's already been digested by the pancreatic lipase, and it moves easily into the simple columnar cell. And then it gets complicated. We can't just leave it at that. Why not? Why can't we just take these fatty acids and monoglycerides and allow them to diffuse into the blood? Why doesn't that work? What's the prominent molecule in the blood? The plasma. It's too easy. Water. And oil and water will mix very well. All right? So we have to coat these proteins with a protein coat so that it can be soluble. Protein has a charge, it's polar, and that interacts with the water charge of the water molecules and allows the protein-coated lipid to be soluble and in the plasma. So here is our simple columnar cell of our lining the villus. Here would be the micelle containing fatty acids and monoglycerol. And it moves into, it's not a membrane bound, it's just this oil separated from the water of the digestive lumen. So it creates its own separation. So that moves in, and we have our to draw out. Monoglyceride and our fatty acids. And that enters into smooth ER. And after all that trouble, within the smooth ER, it recombines and forms triglycerides again. Those triglycerides then leave into and they're covered in a membrane sac because of the smooth ER, and they enter into the Golgi.
Small triglycerides become what?
So the fatty acids and monoglycerides recombine to form triglycerides in the smooth ER, and the triglycerides enter into the Golgi and are now bound by a protein coat.
before they get in the Golgi, right?
while they're in the Golgi.
So what's the coat that's around them before they get in?
They're not, they're just free lipids. So this is just an oil droplet.
What you said, was the membrane sac or?
So yeah, this is just the membrane of the smoothie arm.
Okay.
Okay? So now that fuses with the Golgi, and the Golgi has these proteins that coat, so like low density lipoprotein and very low density lipoprotein, you've heard of those, that coats the triglycerides. And then this buds, this chylomicron, and I'll label it in a moment, buds off of the Golgi. And by buds, I mean it's now in a plasma membrane of Golgi. So that's the Golgi secretory vesicle. Here's the protein coat. Here's the triglycerides. And so this whole structure, not the secretory vesicle, but this whole structure is identified as a chylomicron. The protein coat and the triglycerides is the chylomicron. So once we add the protein coat, that's what makes it the chylomicron. Now the chylomicron, right here, that is too big to go into our vascular capillaries. It's too big to even diffuse across the basal membrane of our cell. And so it's going to fuse with the basal membrane of the cell and be released out into the interstitial space. Too big to get into the capillary. So where does it go? The lacteal. Remember the lacteal cells will spread apart, and that's how they can take up bacteria or large cells, white blood cells. And so this chylomicron, instead of going into the capillaries, ends up in the lacteal. So the lacteal has a high level. In fact, I might have mentioned when we were first talking about the lymphatic system that when they fed dogs a fatty meal, their cisternochyli and thoracic ducts were almost yellow with this milky, creamy substance of chylomicron. And where does the cisternochyli go? Lymphatic fluid that's in the cisternochyli is going to flow into the thoracic duct, which empties into the left subclavian just before it becomes a brachiocephalic vein. And then that flows into the heart. Gets sent to the lungs and back, out the aorta, down the abdominal aorta, and it gets to the liver via the hepatic artery. So we have those two vessels going into the liver that we talked about on Tuesday. Portal vein and hepatic artery. Which one is going to be rich in lipids? Hepatic artery. Portal vein is going to be rich in glucose and fructose and amino acids because those went into the vascular capillaries, into the superior mesenteric vein, into the portal vein, and then joins the lipids via with the hepatic artery. Okay? So portal vein is rich in simple sugars and amino acids. Hepatic artery is rich in lipids. And then those mix together and come out the inferior vena cava and back to the heart again. And then the rest of the body gets the nutrients, the amino acids, and the carbohydrates that went to the liver first. Alrighty, that's kind of a natural break. It's a little earlier than I usually let you guys break, about an hour since we started. But it's kind of a natural break, so let's stop here, just maybe, we've got time. We'll start right at 2 o'clock and do the large intestine. Okay? That's just anatomy, just a little bit of absorption, but no digestion takes place in the large intestine.